Welcome to Revolution Radio, the most disruptive channel on the planet, rebooting, reimagining, and revolutionizing the health and wellness scene, bringing back your grandmother's wisdom without the misogyny, racism, and homophobia. So I want to welcome everybody to Revolution Radio, and today we have Dr. Thomas Seyfried on the show. I'm very excited to have you. I've been following you for a long time. You're somebody who I think is one of the most important voices that we can hear about our health today. Welcome. I'd love you to introduce yourself and just tell us about yourself.
Yeah, well, thank you, Joel. It's a pleasure to be here today. Well, I'm a professor of biology at Boston College. I have been here since 1985, so 35 years. Before that, I was at Yale University for 9 years. I was a postdoc and then on the faculty at Yale in the Department of Neurology. And then before that, I was at Urbana, Illinois, where I received my doctorate degree at the University of Illinois in biochemistry and genetics. So we started working on mouse genetics. I got my degree in genetics. And then biochemistry of gangliosides, which are lipids that are part of the brain mostly. You see them stored in certain diseases like Tay-Sachs disease and these kinds of diseases. It's an inborn error of metabolism, but we were investigating the structure and function of these molecules. And then it kind of morphed into epilepsy when I was at neurology at Yale. Most of the faculty were working on epilepsy. So we did a lot of work on mapping genes for epilepsy with the hope of improving outcome in patients. We had a lot of mouse models of epilepsy that we mapped the genes. And then it became clear that many of these seizure disorders could be managed with ketogenic diets. So, we did a lot of work on developing mouse models for ketogenic therapy, and showed that regardless of what kind of seizure disorder it was, this ketogenic diet had significant impact on managing seizures and all kinds of seizures. So it didn't really make any difference what the gene defect was. The diet was able to manage the seizure. So we became more interested in how the diet was managing epilepsy than in the actual genetic mechanisms responsible for the disease.
Sure. How long ago was this that you
So
started?
that was, well, I started at Yale. So, and we were, still off and on working on that. So this probably started big time in the late 80s, early 90s. But interestingly enough, when I was at Yale, I tried to get an internal grant through the medical school to study ketogenic diet. And they said, "Oh, don't waste your time on that. It's kind of passé." It was a big thing in the 1920s, but we have all these new drugs that work much better, and so I didn't think much of it until I get to Boston College. 15 years later, one of my students said, "Hey, this ketogenic diet looks interesting. Can I go to a meeting in Washington, the state of Washington, Seattle? There's a big thing going on out there." Jim Abrams was there of the Charlie Foundation. He got everybody all excited about the ketogenic diet. So she came back, my student Mariana came back, said, "Oh, we got to work on this ketogenic diet." And I said, "Yeah, I don't know how exciting it is. People think at Yale, they think it's nothing, So I said, we started getting into it based force on one of my students. And then it became clear that the ketogenic diet for managing epilepsy works by calorie restriction. So we could get the exact same results as long as we restricted the diet. And if you don't restrict the diet, we weren't able to manage the seizures. So it became calorie restriction is basically what it is to manage seizures. And we knew that because water-only fasting will stop seizures as well as these ketogenic diets.
Was there a difference between a carbohydrate-rich diet and a ketogenic diet? Was one more powerful than the other at reduction?
Well, if you ate both diets ad libitum, eating all you want, there was zero effect. And John Freeman, the late John Freeman, who was one of the pioneers in the ketogenic diet, he told me, he says, every now and then we get a kid with epilepsy that really loves to eat all this fat, and they never have seizures managed. It's only when you restrict the diet that you really get management. And at the same time, we had a big parallel research program in cancer. And we were looking at lipid biochemistry and angiogenesis and some of the basic fundamental aspects of cancer. And then it just happened that the two research projects overlapped. Why don't we evaluate? As a matter of fact, we had a drug that someone gave us, a company gave us, said, oh, this drug, you might get a lot of effects from this drug. It was an anti-ganglioside drug, actually. It blocked ganglioside biosynthesis. And we gave it to the mice that had tumors, and we said, whoa, look at this drug, works really well. It restricts the tumor growth. But then we found out the mice who got the drug ate so much less food. And then when we took another group of mice with the same tumor and just restricted their food intake to the same level that this blockbuster drug, you got the same results. So the results had nothing to do with the drug. It had everything to do with the amount of food restriction. And that got us onto the idea that what the hell is going on with this food restriction? It works just like stopping epilepsy. It stops the cancer. And then we found out that when you do this to a mouse, it's like a water-only fast for a human. It's like drinking water only. It's like therapeutic fasting. And what happens is the blood sugar goes down and the ketones go up. This is a natural response to the absence of food. And that's when we ran into the work of Otto Warburg, who said that the cancers are driven by glucose. And mitochondria defective, so we knew that you needed a good mitochondria to metabolize ketone bodies. So the cancer cells can't metabolize ketone bodies, and they're surviving on the glucose.
Right. So I want to slow down just a bit, just to make it clear. So Otto Warburg, basically, this was the assumption. We for my listeners, you're one of a few different experts, so that my listeners are aware of a ketogenic diet, and I'm really focused on the mitochondria. How I sort of was alerted to this is I started hearing about these oncogenes, and I said to myself, well, what, an oncogene, is that a gene that is causing cancer? Because, this isn't well known, but is that something, when you hear oncogenes, those are the genes that are upregulated to be able to burn sugar? Is that what it is? Or?
Well, in a sense, you're right. We call them facilitators. So when the mitochondria become damaged from any number of provocative events, if the mitochondria, that's the energy for the cell, oxidative phosphorylation. So if that process becomes damaged, acutely, the cell will die, and you'll never get a cancer cell from a dead cell. But if the damage is chronic over, extended periods of time, it gives the cell an opportunity to shift its metabolism from oxidative phosphorylation to a fermentation, which doesn't require oxygen. And the oncogenes are facilitators of the transition from respiration to fermentation. So they control pathways that allow the cells to take in more glucose and glutamine and metabolize these two fuels through these ancient metabolic pathways. So the oncogenes are essentially facilitators. They're not the cause of the cancer. They're facilitating the transition from one form to another. If you take the cell that's already neoplastic, that is cancerous, with the mutations and the oncogenes and all this, and you put it back into a new cytoplasm with new mitochondria, all those facilitators turn off, and the cell now restores its growth regulation and becomes a differentiated cell again, despite the continued presence of the genetic defects.
Right.
So the oncogenes are just responders to the transit. They facilitate the way the cell, because you got to open up the floodgates to bring in fermentable fuels. And that's what the oncogenes do. HIF1 alpha, MYC, RAF, all these kinds of things.
Okay. Well, I want to just give a real quick basic understanding of, how you would explain cancer, just so people can understand that, this is something that you're very used to talking about. I've heard you speak many times. And for people hearing this for the first time, this is kind of a shocking statement that, everyone thinks that cancer is genetic. Everyone will tell me, I had an uncle with this, with melanoma. And, they're talking along the lines that cancer is a genetic disease and that we don't know what the root cause is. So just real briefly, how would you describe what cancer is just to the average person?
I think that's probably one of the greatest tragedies in the history of medicine. No, bar none. I've said it before. the misunderstanding of cancer as a genetic disease will in time become recognized as the single greatest tragedy. In other words, you had a false understanding of what the nature of the disease actually is, and it still perpetuates today. So, you need to know that Otto Warburg was in fact correct when he defined cancer as a disease of energy metabolism involving defective mitochondria. And a lot of resistance in the academic, well, there's a lot of data that provides misinformation and makes it look like, cancer cells have normal respiration, but that's not true when you really look at the data. But what a tumor cell is, essentially, what cancer is, are simply a group of cells growing out of control. They have lost their ability to regulate growth, so they continue to proliferate, and then of course it destabilizes the local tissue microenvironment, leading to an inflammatory kind of condition. And where metastasis comes from is basically we have immune cells in our body, and whenever our tissues are damaged, like from a cut or a wound or some contusion or whatever, we have macrophages. These are cells that are part of the immune system, and they facilitate wound healing. They're kind of facilitators. They can reconfigure the damaged microenvironment. They can rebuild new blood vessels. They can do a lot of things to reconfigure tissue damage. But when the tissue damage is chronic, these cells can fuse. Like if you have some cells that are proliferating, but they can't spread around, they just kind of proliferate in place, and they create a disturbance, like a wound that does not heal. So our immune cells come in to try to heal this wound, and in the process of trying to heal the wound, they spew out growth factors that are supposed to they're supposed to help wound healing. But they act like gasoline on these incipient cancer cells. So they grow faster, and then the cells fuse. This macrophage will fuse with one of these proliferating kind of stem cells. And then you have this kind of hybrid cell. Now, this cell is already genetically programmed to move around your body because it acts like a militia in the body. If you find a disturbance, they come out of the bloodstream, rush into a particular area, and try to facilitate wound healing. And we also have the local police department. These are macrophages that actually reside in the tissues themselves, and they're designed to handle very small incidents of tissue damage and repair and replacement. Well, those cells in an incipient cancer, they can also fuse with these cells. So you get kind of a lot of cells programmed to do what they do in a wrong context. And then it leads to an escalating situation, a biological chaos. So you have all these cells basically screaming at each other metabolically, and it leads to chaos, fusion hybridization, and then you have these cells that spread around and become what they call metastatic cancer cells. But basically, they're part of our tissues that have become genetically unstable, leading to proliferation dependent on fermentation fuel. So when you look at the evolution of species on the planet, before oxygen came into the atmosphere some 2.5 billion years ago, all the organisms that existed on the planet then were all fermenters because there was no oxygen. So these pathways of energy metabolism through fermentation are ancient, very ancient pathways, because all the organisms that existed before oxygen were all fermenters. So when oxygen came into the environment, there were certain cells that could trap this oxygen and use it to make energy. And these cells then became some sort of a hybrid also. And the mitochondria are a bacteria that were able to capture oxygen, and they fused with another kind of cell, and they became metazoan cells, which are eventually us today. So in our bodies, when we have cancer, these cells are falling back on these ancient pathways. And during that time, 2.5 billion years ago, all the organisms on the planet were highly proliferative. They proliferated like crazy as long as the fermentable fuels in the environment were there. And when the fermentable fuels were depleted, these organisms would simply die. So the cancer cell is falling back on these ancient pathways and will grow uncontrolled as long as there are fermentable fuels in the environment. And those two fuels now are glucose and glutamine. We have identified the two fuels that drive the beast.
Right. Okay. So at this point, what I want to make clear, we talk a lot. I talk a lot about the mitochondria. So at that point in history, now we're utilizing oxygen. We have oxidative phosphorylation, and was that the key to multicellularity? Is that what we needed? Because
yes.
Okay.
Yes, because that allowed cells to sort themselves to do divisional. Each cell would have a different division, separate different functions. Right. So metazoans, you move from single cell organisms to metazoans, which are multicellular organisms.
Right.
And then these cells could take on specific function, a division of labor, so to speak, and that becomes the state of differentiation. And now we have the ultimate in all modern organisms that exist on the planet, mammals. I mean, there is a division of labor. Kidney cells do one thing, liver cells do another, neurons do another. And the mitochondria are present in all these different kinds of cells. And they all regulate the energy metabolism in these different kinds of cells. So when some provocative agent, like an inflammatory condition, a chemical carcinogen, if you will, viral infection, if you will, whatever it is, would damage the mitochondria in a particular cell, that cell will either die or, if the damage is not so acute, can upregulate a fermentation metabolism driven by these facilitating oncogenes.
Okay.
Now, what happens then is the mitochondria throw out these reactive oxygen species. They're radicals, because their respiration becomes defective. And the radical reactive oxygen species are carcinogenic and mutagenic. So the somatic mutations that you see in the cancer cells are coming as the result, downstream effect of these reactive oxygen species. Yet the field continues to focus on all these these mutations, thinking that they're important.
Right.
They're actually downstream epiphenomena, absolutely unrelated to the cause of the disease.
Right. Okay. So I want to, before we get into that, there's lots of different things I want to talk to you about, because I just wanted to clear up the science for people. Because when I start to talk about this, a lot, I've been seeing patients here for 20 years. And, I love my patients. They've taught me everything about life, but they're getting such a completely different message. So a lot of them are worried about, the causes of cancer. So basically what you're saying is that there isn't one cause of cancer. There's some assault to our body, our cells, and there becomes damage in some way, and it's the mitochondria that gets damaged. And as a result of the mitochondria breaking down, the DNA becomes distorted. And unfortunately, medicine is focusing on the distorted DNA rather than the root cause, which is the actual production of energy. And the fumes coming out of the mitochondria are really corroding the area, and this is causing the disease.
Yeah, and in so many words, that's true. And you can cause that problem. Like, for example, there are oncogenic viruses, papillomaviruses. You have the what is it? The liver virus there, hepatitis virus. There are a variety. Those viruses, either the virus themselves or the products, blow out the mitochondria in a chronic way, and therefore you get liver cancer, you get papillomas, you get all these kinds of things, and you have. If you have an occlusion in a particular tissue where you have a hypoxic region, a little bit of a hypoxic region that gets, like the oxygen would come in, cause mitochondrial damage, then it would become hypoxic. It's back and forth, and eventually you end up with the cell that has damaged respiration and has transitioned into a fermentation metabolism. And some of our, you can have genetic risk factors as well, but they're no different than a chemical carcinogen.
Right. Okay. Well, so you basically led me right down to the path that I wanted you to, because I treat sleep apnea, and that's how I got to you in the first place. Is that in my investigation of sleep apnea, it came to me directly that this was a metabolic disease. And so when you're saying this hypoxia, sleep apnea is basically stopping your breathing. I know we sort of spoke briefly, and you knew right away. You said, well, sleep apnea, that would potentially amplify cancer. So based on what chronic intermittent hypoxia, which is what sleep apnea is, what's the effect on cancer, in your opinion?
Well, if the body is inflamed, you see, it's more than, if you have inflammation, can cause cancer because it damages mitochondria. It's an inflammatory situation. So if the body is naturally inflamed, measured by C-reactive protein, people can, every time I go to get a blood work, I always say, what's my CRP? They say, why you want to know that? Because I want to know if my body is inflamed or not. puts me at risk for a
Right.
lot of different conditions.
Right.
So if, obesity is often, not always, associated with inflammation. Sleep apnea can happen more frequently in people that are obese in general, not always. So you put an inflamed body together with intermittent hypoxia, and Otto Warburg clearly said intermittent hypoxia is the origin of many cancers. So you put your body at risk, and who knows what organ, by not breathing and being inflamed, it could come out with a breast tumor, a prostate cancer, a colon cancer. It doesn't make any difference. Whatever it is, you put your body at risk for two provocative insults, which is intermittent hypoxia together with inflammation, and you put yourself at significant risk for developing a tumor.
Right. Okay.
It's not that complicated.
Right. Okay. Well, listen, I've been speaking about this for a while. So I want to just clarify one more thing before we talk about a sort of a different idea here. And that, so as a researcher, as a doctor, you're very well read and well traveled in the biologic areas here. So to me, when I heard a lot of what you said we took the cancerous nucleus and put it into a fresh cytoplasm, the cancer disappears, and we took a good DNA and put it into a damaged cytoplasm and cancer happened. So is that, when people are listening to this, not to be too sciencey, but, people know enough because we're seeing these commercials on TV for gene targeted therapies, and they're really bothering me because they're showing these real people and saying, I had this blah, blah, blah gene targeted therapy and it's, saved my life. Maybe if you could just simplify this just maybe one more time for anyone listening to this, that this is, how did you personally come to the conclusion this is not a genetic disease? Did you wake up one day and say, you know what, I've been researching this and that's it. It's just not a genetic disease. This is just definitely a metabolic disease. Was there any?
Yeah, well, it didn't come, it came from the nuclear transfer experiments first, I think. I had a lot of evidence that Warburg was correct because I did work. My group isolated mitochondria from cancer cells, and we looked at them very carefully, and we found all kinds of abnormalities at the biochemical level. They were just damaged, but there was no genetic damage in the mitochondrial DNA in the cells. So I said, "What the hell's going on here?" But the cardiolipin, the lipid inside the cristae of the mitochondria, was defective. And then we looked at the actual enzymatic activity of oxidative phosphorylation complexes, and they were abnormal. So clearly, these cancer cells were not respiring effectively. So I knew intrinsically that cancer was a mitochondrial disease, and I actually read carefully all of Warburg's work, and even some of the German translations, and I came to the conclusion that he was correct, but. There had been trickles of information over decades that looked at cancer from a developmental perspective, not even challenging or recognizing, for the most part, Warburg's theory of cancer, and just asking, does a cancer nucleus, from the nucleus from a tumor cell, can it direct normal development? And people were expecting that it would not be able to direct normal development. So they took nuclei out of tumor cells and put them into new eggs or new stem cells, and they were able to develop a mouse or a frog or something from the nucleus of a tumor cell. And I said, what's going on with that? Because according to the somatic mutation theory, these things should have been dysregulated in their growth if genes really do that. So what I did, these guys were all very distinguished developmental biologists, by the way, some of the leaders in the world and still remain that way. Rudy Janisch at MIT is considered one of the foremost leaders in mammalian developmental biology. But these guys were not asking the question, do these experiments support or reject the somatic mutation theory of cancer? So what I did is I reevaluated all of these different kinds of studies in frogs and mice and various kinds of human fusion cells and all this, and put it all together in one paper, linking it all that these studies, viewed in a different light, directly refute the gene theory of cancer. So when you look at that, and then you say, wow, no one has yet been able to argue against the nuclear transfer experiments. Then that led me to further investigations where did this idea come from in the first place? Who first said cancer is a genetic disease? Who is this guy? And turned out it was Theodore Boveri, a very prominent German scientist from 1914. He was famous for, actually, Boveri was famous for linking Gregor Mendel's abstract traits to chromosomes, because you have to realize genetics emerged in 1900 as a new science, mainly by finding that there was a biological basis for Gregor Mendel's abstract traits. So Boveri was one of the first to say that these factors that Mendel were studying was on chromosomes. But then Boveri, I don't know what motivated this guy. He started to say that, well, let's think about cancer. And basically, he said he thought cancer might be something to do with chromosomes, but he was extremely apologetic about it. He says, listen, he told in his clearest paper, I don't know anything about cancer, never studied cancer. What I'm doing is purely speculation, and please, give me some slack here. I'm probably wrong. And he was very apologetic for his total lack of knowledge about cancer. Now he's becomes the founding father of the whole order, who knew nothing about cancer. So now then we, with the deep sequencing, many years later, where you can sequence thousands and thousands of genes. Well, it turns out the number of scientists have found cancer that have no mutations. No matter how hard they look, they can't find any mutations. Well, how can you have a theory based on mutation when you don't have any mutation? Yet you have a cancer cell with no mutations. Well, we don't talk about that. And now the recent studies that have just come out in the last couple of years, we're finding all these driver genes. A driver gene was defined by Burton Vogelstein and others as being the kinds of mutations that are actually responsible for the origin of the disease. And now we find normal tissues that have driver gene mutations and never develop cancer. So we have cancers with no mutations and normal tissues with driver mutations and no cancer. And this is all incomprehensible for the somatic mutation theory of cancer. Right. And then you have which is really interesting, is we go back and look at organisms that are like us genetically. And the chimpanzee is one of the closest relatives we have, 98.5% similar in gene sequence and protein sequences. And those guys rarely, if ever, get cancer. There's never been a documented case of breast cancer in a female chimpanzee, and they're almost genetically the same as us. So what the hell's going on with these guys? Why they don't have any cancer, and humans are loaded with cancer?
Right. Right. Okay. Well, okay. So, you don't know my background, but I do talk a lot about epigenetics, and for me, this is a very passionate project that I have based on my life experience. But I want to sort of switch gears here and say, okay, so basically, I feel like you're qualified to say, hey guys, we're looking down the road here. Cancer is different. Now, when it comes to the real world, here you are. You're sharing this message. You've been sharing this for a while. Is anyone listening, and are you having any impact? I know that America can be very hard because of how things are structured, but are you having any effect? Is your information, or do you get in trouble where you are for being so contrarian, or what's going on? Are people, is there any change that you're affecting with this information?
Well, I think the information is seeping out for people that are willing and able to look at the data. Okay, so and I have to be honest with you. Anytime you have a major paradigm change, there are people who just refuse to acknowledge it. So the reason we don't have a more a general discussion and understanding is for several reasons. One is that simply, like you say, a lot of people never heard of this. All right, so it's a lack of familiarity. It's lack of knowledge, unfamiliar. I never heard of this stuff. Where did this come from? All the textbooks say something different. Okay, well, the textbooks are not up to date on the scientific literature in a lot of cases, right? Right. And then you have the power of ideology. Ideology is stronger than any gene that I know of. I mean, people are indoctrinated by religions and by political persuasions. I mean, you know how hard it is to break an ideology. So if you've been indoctrinated effectively by either a religion or a political affiliation or whatever, there's nothing you're going to do to change this guy's mind. Yep. So that's ideological resistance. Yep. And then of course you have a lot of people in the industry whose jobs depend on them not accepting this. They can't accept it because they're working on a totally different thing. You put all three of those together, and that pretty much encapsulates the failure for this concept to be more widespread, and recognized.
Right. So what I want to, just let you know is that, so that's where I'm at with the whole epidemic of sleep apnea. And you said some very important things. Apnea is associated with obesity, there's no doubt. But as a clinician, my latest patient was a 23-year-old petite female. And so to me, this is not a surprise because that's who I'm seeing now. When I learned how to treat sleep apnea, I was looking for the obese person with a large neck.
Right.
And the paradigm is impossible. So I have a different theory. And when I went into my neighborhood, my very hardworking dentist, they have this obesity image in their mind. And when I was taking classes, they say, well, you're going to measure the person's neck. And if they have a big neck, then they probably have apnea. So this is one of the hardest things that we can talk about to change these ideologies. But I want to let you know that I'm coming from exactly the same background, and it's the same paradigm. Sleep apnea is a metabolic disease, same as cancer. So we're looking at the same disease. I think we're looking at the same disease, just from different views. So having said all that, there's also this giant fear that when someone gets diagnosed with cancer, they just drop everything and they go, I've got to go get this out of my body, get chemotherapy or cut it out or whatever. And so there's a giant fear factor as well. Both of my parents passed from pancreatic cancer, and I saw what happened. So for your, if you were in charge of research in America and you had some say, what would you change about the current cancer industry, just in general? Because, people, these systems are very ingrained in our system as a standard of care, and anyone who goes against them is called out for many different things, including, being a quack. And that's the issue that I have, is that this industry is not going to be, it's just not easy to change people's opinion. What would you change? How would you like to see this, in the industry? Because you're working in this.
Yeah, well, I mean, knowing what we know about the origin of cancer and why the cells are proliferating out of control, I mean, standards of care, radiation and various chemos and these kinds of things, they're all designed to stop cell proliferation, basically. I mean, you're going to irradiate cells, you're going to break the DNA, you're going to try to stop them from proliferating by creating reactive oxygen species in the microenvironment to break DNA. A lot of the chemotherapies are involved with binding up microtubules or stopping DNA replication, or everything is to stop the proliferation of the cell. But you have to realize no cell can grow without energy. And the energy that's coming to drive the cell is purely fermentation energy. So you realize that what are the two fuels that are predominantly driving fermentation? It's glucose and glutamine. And we find, and others and many people have found, no cancer cell can live without glucose and glutamine. So the strategy to manage cancer becomes extremely simple. It's not complex. Now, every cancer is the same. Whether you have a brain cancer, pancreatic cancer, prostate cancer, colon cancer, bladder cancer, they're all fermenters. It just happens to happen in one tissue or another tissue. So if you know that, then the strategy becomes, how do we stop glucose and glutamine without harming the rest of the body? So that's why we bring the patients into therapeutic ketosis. So they transition away from the sugar, the glucose, and then we target with drugs and procedures to target the glutamine and the glucose simultaneously without harming the rest of the body. So, the goal is how do we manage the disease without harming the patient? And and if we know that the cell can't live without two fuels, then the strategy becomes crystal clear what you do. So the question is, why is that not being done? Why is the National Cancer Institute and all the pharmaceutical agents? Because their current protocols and treatment strategies are based on extremely expensive drugs and procedures that generate massive revenue. So you're not going to be able, but the bottom line here is that this is not an insignificant thing, because we're sacrificed as the result of not moving in this direction. We essentially sacrifice 1,600 people a day dying from cancer, because we don't want to move in the direction that we need to move into.
Right.
Well, this is the tragedy. The tragedy is 1,600 people a day in the United States dying from cancer. In China, it's 8,100 a day dying from cancer. And we bring these poor souls into these clinics, and we irradiate and poison them, and with the hope of stopping cell proliferation. Hair falls out, people get sick as a dog, all these kinds of problems, and that doesn't have to happen. And people will say, well, how come this isn't known? How come you're not doing this? Because no one has yet found out how to generate revenue with this. And cancer is a revenue generating disease.
Yeah.
So there you have it. You'll go back to the same three things. I'm unfamiliar. I'm an ideologue. Oh, my job that prevents me from doing this. All three of those things happen.
Okay, and so thank you for saying that, because that's a big theme of what I talk about in my book. Now, I'm Canadian, and I come from a more socialistic system. It's completely different. So what you just said to me, I've been living in the U.S. for 20 years, and it's obvious to me that these drivers of this whole industry, it's very hard to stop these powerful industries. But why do you think something like this can't take root more in a country like Canada? Because these diseases are going to bankrupt all of us, because I don't know if people figure this out yet, but this is how people die these days. They die from heart disease, from cancer, or neurodegenerative disease, which are technically, in my opinion, all mitochondrial diseases. But so what do you think? I wanted also, the same question is, are there other countries that you have collaborated with where they're starting to say, hey, just because the industry goes this way, can we buck the trend? What can we do? Tell me, is there anything you feel hopeful?
I think, I've worked with a lot of cancer patients from Canada and a lot of physicians. The socialistic system is locked as hard, or maybe even more into the standards of care. In fact, when it becomes government approved, it becomes even harder to change the damn standard of care.
Yeah.
So I work with the English, the Germans, the Canadians. They are so locked into this socialized standard of care that you need an atomic bomb to change it. So, and I think if we go into a socialized medicine, we're going to be locked into radiating and poisoning people to make them healthy. Can you believe this?
I can. Well, they talk about putting cholesterol statins in water. I want to sort of, now that we're talking about a general healthcare issue, what's your opinion on cholesterol statins? How do they fit into what you think? Is this related to cancer? Do you think cholesterol statins are safe? Are they a good thing? And should they be put in the water supply, like some people are saying?
No, I mean, David Diamond has clearly shown, and others have shown, that these statins don't do anything. They can, they're not. Yeah, it's another thing. It's like red herring. You're looking at cholesterol. Triglyceride levels are more important for heart disease than our cholesterol levels. You have to look at these things, and this is another thing that, that the industry has found to make everybody fearful of cholesterol. And therefore, it's another thing. It's just you need to educate people, but you have these big forces in place, like you've mentioned, that prevent the information from getting out. And even if it gets out and it's contrary to what your institution believes, you'll be vilified. The issue here, in our case, is when you're standing on a bedrock of solid knowledge. It's hard to challenge that when you have such a massive amount of evidence supporting what you're saying. So it's just a matter of time. And besides, with cholesterol and this kind of thing, if you might have a heart issue. Well, you're not going to die, a miserable death in 3 weeks from this. But with cancer, these people are being tortured before they die. I mean, this is terrible. I mean, not only do you have a disease that's potentially life-threatening, but many of the treatments can actually provoke it. Some of the new immunotherapies that we have, the so-called PD-L1 inhibitors that you hear advertised as being the new, they can make some lung cancers grow twice as fast.
So they have these people on TV, on commercials, saying, I did the PD-L1 pathway.
Yeah.
So this sort of leads me to the same discussion, is that a lot of these diseases are diseases of communication rather than diseases of science. Any real scientists who will look into this will see what you saw, basically. So then I guess the question is, how do we change the messaging about this? Is there you have any thoughts of what could we do to help share this message? And what I'm doing right here, right now, just so you know, is I believe your information is so critical to health, and I watched my parents die, I watched what their doctors did, and I want to share your message with as many people as possible. And you've been thinking about this for a long time, and I hear your frustration, and I agree with you. But what do you think? Do you have any thoughts? And I'm trying to bring this information out in a completely different way. I'm trying to use much more of an entertainment, infotainment type of focus. But what do you think? What do you think?
Well, I think the momentum for the change will have to come from the bottom, not the top. And I think the bottom line is people want to live. And I can't tell you how many calls I get from cancer patients and physicians, the likes of which, the bottom line is that the patients, once they listen, people aren't stupid. They go on the internet and they start looking at these things, and they see people that are living who had various stage 4 cancers, who are doing pretty well. And they're saying, what did you do? Why? So they'll run to their oncology center and say, I want metabolic therapy. And the poor guys will say, well, I never heard of that. If it were any good, I would have heard about it. No, you wouldn't. You wouldn't hear at all about it. The medical students aren't even trained to know anything about it. So it's going to have to come from the consumer, and the sick patient is your consumer. Yeah. And if the consumer says, I want metabolic therapy and instead of chemo and radiation, all of a sudden these guys are going to have to change their tune. So, once we have enough case reports written, and I'm working with physicians all the time, writing up individual case reports on people who have used metabolic therapy as an alternative, not as a complementary, as an alternative to standards of care. And they seem to do better, live longer, have a higher quality of life. What's wrong with that? What's wrong with it?
Oh my God. Okay. Well, we're going to be for sure posting your contact information so people will be able to find you. And, hopefully if things go well and everything after launch my book, I'd love to, maybe do another interview. I really appreciate your time. Thank you very much.
Well, thank you. I hope it helps you out and your cartoon characters and everything else.
All right. Absolutely. Well, you got to do what you got to do.
Yeah, you do. All right, Joel, listen, I'll let you go.
Thank you very much.
Thank you very much too. Bye now.
Have a good day.